So now that we understand how we make that recombinant plasmid, we call that recombinant plasmid a vector. In gene cloning, the original plasmid is called a cloning vector. A cloning vector is a DNA molecule that can carry foreign DNA into a cell and replicate there. So back on this picture here we made the recombinant DNA. This serves as a vector, a way to get that human gene into a bacteria cell. Okay, so producing clones of cells. Cloning a human gene in a bacterial plasmid can be divided into 6 steps. The first one, the vector and gene source DNA are isolated. So that's where this is. That's back to this picture here. So we isolate the bacterial plasmid and we isolate the human DNA. The DNA is inserted into the vector. So that's what this was here. They were inserted into the vector, and this was the nitty-gritty as far as how we do that with the use of restriction enzymes. The human DNA fragments are mixed with the cut plasmids, and base pairing takes place. So you have your cut plasmid here. We mix it with the cut-out human gene here, and then they hydrogen bond, and we have that base pairing take place to be able to make the recombinant DNA. 4, the recombinant plasmids are then mixed with bacteria. So now that we have the recombinant plasmids, we have to mix it with the bacteria in hopes that the bacteria will take up that recombinant plasmid and therefore obtain a human gene. Then, so we mix the plasmids with the bacteria. Remember, these are really small. We can't see it happening. So we have to just mix it in solution and hope that the bacteria took it up. So then we plate the bacteria on a petri dish and incubate it so it's nice and warm and good conditions to grow and divide. And then we have to identify the clones with the right gene. We have to figure out of this bacteria growing, what bacteria, which of all these different bacteria actually have the human insulin gene. Okay, so that is what this picture here is showing you here. So we're going to get a little bit more detailed here. So the first step was isolating the plasmid. So here's the plasmid. Now notice here a couple of things about the plasmid. The plasmid has, right here, I'm going to color it orange. Right here is AMP-R gene, ampicillin resistance. Ampicillin is an antibiotic. So having this gene makes this bacteria and the ampicillin resistant, and so therefore when you grow it on a petri dish with ampicillin in it, it won't die. The second gene here that I want to point out is this gene here, which is the LAC-Z gene. So right here is the LAC-Z gene. This is codes for making an enzyme that digests lactose breakdown. But notice, one thing I want you to notice here is the little jagged line in between here. So right here, there's a little jagged line right smack dab in the middle of that lactose gene. That's a restriction enzyme cutting site. All right, that's the restriction site. So remember, the first step is to isolate the plasmid. So we picked that plasmid. So all of these plasmids have that ampicillin resistant gene. But when we use the restriction enzyme to cut it, the restriction enzyme cuts smack dab in the middle of that LAC-Z gene. So is the LAC-Z gene going to work anymore if we put a gene in between here? No, because it won't be intact anymore. So that's what we've done with the bacterial plasmid. Then we look at the human DNA. The human DNA here, we take the whole chromosome and we cut it into fragments here. So you have this black fragment, and then I'll color this one blue, and this one, what color? Let's see, green. We'll use green. And this one green, so we have black and blue and green. And only one of these fragments, so you use this restriction enzyme and cut up the DNA, but there are lots of fragments there, and the only one is the black one that actually has the gene of interest. These other two don't have our human insulin gene. But when we're doing this in the laboratory, all of this is happening in like a little test tube, and you can't see the DNA, you can't see the restriction enzymes, and you for sure can't see which gene or which fragment of the DNA has the human insulin gene in it. So we end up with all these fragments with just one of them containing the gene of interest. So now we've cut up our DNA from humans and from the plasmids with the same restriction enzymes. That's important that we use the same restriction enzymes because now you have the same cutting sites and the same, therefore, sticky ends so that they'll bond together. So that's what we do here next is we mix the DNA, and the DNA will join by base pairing. So when we see here, so this one, let's put some numbers on here. So the one with the gene of interest is DNA number 1, and then this is DNA number 2 and DNA number 3 here. So when we mix them together, some of these plasmids will mix with the human gene 1, which is the gene of interest. So that's what this one has, human gene 1. So that has the gene of interest in it. Let's write gene of interest. Then we have this one. Here are 3. Looks like right here, inserts into here, so that's gene 3. That's not the gene of interest. And then we have this fragment here joining right here, and so that is fragment 2. And so we only have one plasmid with the gene of interest in it, so these are your recombinant plasmids. Another possibility, though, is that as you mix these in the test tube together, it could be possible that these plasmids rehydrogen bond together, and so it could be possible that you end up with a fourth kind of plasmid that has no human DNA in it. That's possible too. These ends can come back together like this. So the difference between this one and this one, all right, they all have, remember the plasmids all had the ampicillin resistant gene. So they all have the ampicillin resistant gene. That's kind of hard to see here, the orange. They all have the ampicillin resistant gene. These ones that have the human gene inserted now have that broken up LAC-Z gene. So they're not going to be able to digest lactose. That gene's not going to work. This one, though, since it doesn't have the human gene, has an intact LAC-Z gene. All right, so there's nothing going on with that. Okay, so now we have these recombinant plasmids, or maybe not, like this one, and we mix them with the bacteria, and we hope that the bacteria take up these plasmids. So what happens is you have some bacteria that take up this plasmid. And so, I don't know which one it is. It's hard to see here. Let's say this one here. So this one takes up plasmid 2. So this is plasmid 2 here. So it has the blue in there. This one takes up 1, plasmid 1. And remember, so this is the gene of interest. And this one, let's say, takes up plasmid 3. So they have all different plasmids. Which is the only one that we're interested in? This one here. This is the only one right here that has the gene of interest. But remember, we also have this other plasmid here. So it is a possibility that you have the bacteria that take up plasmid 4. All right, and then plasmid 4 goes in there, and they don't have any human DNA. And then you also have the possibility that you're going to end up with bacteria that didn't take up any plasmid, so with no plasmids. Because just because you mix the plasmids with the bacteria doesn't necessarily mean that the bacteria is going to take up that plasmid. All right, so these are our possibilities. So the question is that now that we have all of these bacteria, all these different sources, remember they're all sitting in a test tube. We can't see them. So how in the world are we going to know which one has human DNA and which ones don't? Because these guys don't have human DNA and these guys do. And so in comes this last picture here. It says, place the bacteria on agar. That's the gel stuff that you grow it on. Place the bacteria on agar containing ampicillin. So if it has the ampicillin, only the ones with the plasmid is going to grow on it. So because remember the plasmid, so plasmid number 2, they had the ampicillin resistant gene. So this one's going to have the ampicillin resistant gene. This one's going to have the ampicillin resistant gene. This one's going to have the ampicillin resistant gene. But this one right here is not. So by placing ampicillin on here, you're going to kill off any bacteria. You're only going to have bacteria growing that took up a plasmid. All right? And so that's the purpose of the ampicillin. So these guys are going to grow on it. This one is not. Then the second thing is to weed out, how do you know which one has a human gene? Like these 3 have the human gene. This one does not have a human gene. So how do we distinguish between which kind? So that's what this X-gal is. And so let's write along the side here a piece of information. An intact LAC-Z gene produces a functional enzyme that breaks down X-gal, which is a molecule, in the medium that it's growing on, turning it the color blue. So that's going to be important here in looking at this one here. So only the bacteria with an intact LAC-Z gene. What are the bacteria with an intact LAC-Z gene? That is the intact one. Remember all the ones that have the human gene in it? It broke up the LAC-Z gene. So the only one that has an intact LAC-Z gene is this one right here. Right? This one right here. And so if it has an intact LAC-Z gene, when it grows on this medium, it can grow on the ampicillin because it has the ampicillin resistant gene. If it has that intact LAC-Z gene, it's going to use this X-gal in the medium, and it's going to turn blue. So all the dark spots on your Petri dish here, all these dark spots, I'm going to color them blue. These are all blue. These guys are carrying non-recombinant plasmid with intact LAC-Z gene. So that means that these guys here, these are the ones that don't have the human DNA. So we're interested in all the ones that are not blue that have a disrupted LAC-Z gene. So what does it mean if it's a disrupted LAC-Z gene? It means it has a human gene. So now our next task is to figure out, all right, all these ones that are white have a human gene in them. Which one is this one that has the human insulin gene? And that's what we're going to look at next.